you very much for your invitation to have me here today, and hi to everyone. I'm Ehsan Habibi. I'm a postdoc in Fashion at the Broad Institute. And today I'm gonna talk about deciphering the molecular basis of cellular diversification in the mammalian cerebral cortex. The cerebral cortex is the region of the brain responsible for execution of most sophisticated higher order functions, including cognition, sensory perception, and sophisticated motor control and communication. And achieving these all functions relies on a diverse group of specialized cell types with precise spatial organization, including projection neurons that extend their axon to different regions of central nervous system, and they express selective candidates of genes and also other types of cells like glial and interneurons. Over the past decades, and also specifically over the last few years, there has been huge efforts on characterizing and understanding the diversity of the neural cells, specifically in adult cortex via different consortia like BICCN, Brain Cell Atlas, also Human Cell Atlas, using single cell transcriptome. But we know that cortex is a very unique organ. And from developmental biology point of view that I'm very interested in, corticogenesis is a phenomenal model to study how diverse cell types emerge from a homogeneous population of cells through a very tightly regulated series of spatiotemporal events. And then later on, these cell types precisely allocated and integrated into their final destination. So, for instance, during the embryo development, these apical progenitors lining the ventricle zone proliferate asymmetrically to self-renew themselves, and at the same time give rise to intermediate neurons and intermediate progenitors, and finally different types of neurons are generated sequentially over different times, and they populate the cortex. For instance, as you can see here, around day 10.5 or 11.5, you see these symmetric dividing neuroepithelial cells, and over time around 12 and 13, you see that this is the birth date of layer 6 and layer 5 of the excitatory neurons. And later on, around 14 and 17, you can see the birth date of layer 4 or upper layer neurons. And later on, you can see the gliogenesis happens. And by the end, a layered cortex will be formed over time that, like, different types occupying different actually depths of this tissue. Interesting questions that we can ask now is that when is neural identity established, and are these progenitors prefated that actually give rise to different types of neurons, or actually they are not? And then, over time, they process happen. So to understand the sequence and timing of this key transcriptional and epigenetic dynamics that correlates with this emergence of these cell types over time, in a collaboration with Paola Allotta and Daniela Di Bella at Harvard Stem Cell Institute, we generated a very comprehensive map of subcerebral, the neocortex, developing neocortex over time using single cell sequencing, single cell attack-seq, and complemented with Slide-seq. So therefore, we profiled the mouse prospective somatosensory cortex using a single cell sequencing over the entire period of corticogenesis from day 10 day P1 every day, and also P4 as a more adult sample. And here on the left, you can see the UMAP visualization of this individual time points. Each dot is cell, and we annotated based on the markers, known markers that we have, and therefore we define the identities at each time point. And also you can see not only cortical origin cells, but also non-cortical origin cells like interneurons and microglia and vasculature that actually they are coming from the they have a non-cortical origin actually. And on the right, you see the composition of cell types over time. You can see, for instance, during early days, you can see mostly apical progenitors and intermediate progenitors. And over time, you can see the emergence of cell types and specified cell types. So as you can see here, now if we get all these individual time points, merge them all, you can see the main differentiation trajectory is a continuum from apical towards projector neurons and glia cells. And you can see that, for instance, in the earliest stages, you can see the expression of, apical progenitors like SOX2, and also during like these intermediate precursors or progenitors, you can see the expression of EOMs and others. And also later on, you can see the expression of tube tree as a markers for projection neurons and in-general neurons. But something that is interesting here now, we can go through all the cell types and define the signatures over time. And this is important because you can get access to all markers that we might be interested during these time courses. So as you can see here, that this is a spatiotemporal event. But when you do single cell, you remove the positional information. And we know that this is an important aspect of corticogenesis. So therefore, we took the advantage of spatial transcriptome methods that has been developed in Phase and Evan McCaskill lab, Slide-seq, that you already heard about that, to associate cell identities that we defined from single cell transcriptome to map them back to their topographic organization. Here we used a Slide-seq, which is approximately 10 micron resolution. And we actually later used Tangram, a deep learning approach that has been developed in Abir's lab, which uses actually single cell data that we defined and annotated, and also take the spatial profiling data from the same region with the same age or tissue to map the cell types to their tissue. So just briefly, Tangram first, randomly placed the single cell profile in a space and then compute an objective function that mimics the spatial correlation between each gene in single cell and a spatial data. So on the left, you can see the learned spatial distribution. You can see nicely that different cell types, deep layer and upper layer, has been actually placed on their expected position. And also you can see as early as E13.5, you can see nicely we can project that apical projection neurons and migrating neurons, and also later on during E15.5. One of the interesting observations from this spatial data was migrating neurons, which are actually transient cell types. And migrating neurons leave the ventricle zone, travel to the cortical plate to reach their destination. And when we look at our single cell data, we see there is a heterogeneous population with several distinguishing clusters. And we used Tangram actually to map them back to their, topographic place. And then you can see that nicely there is a gradient of cells. So each population has been mapped actually to different places in the spatial data. That means that migrating neurons changing their transcriptome profile, where they just migrate through their cortex actually over time. So now we can associate these states and with differential gene expression, and we show some genes that exhibit differential expression both in the single cell data and in the Slide-seq data, and are actually sequentially expressed during migration of these neurons, and indicating that a spatial gradient actually appears encoded in an expression gradient in these cells. I need to emphasize another thing, and that is actually when you look at single cell data is what we collected is discrete, because we just sampled every day. And although we kind of covered every day of this process, but it still is a discrete. So therefore, to study the differentiation continuum, we can assign each cell a numeric value that measures their position in a dynamic process of interest, here like differentiation. And this value called pseudo-time value, which is in accordance nicely with the collection of the data. You can see here, for instance, from day 10, day 11, and 12, and along the time, you can see the emergence of various cell types and neurons. And actually, the pseudo-time value captures very well the time of collection, showing that the transcriptional profile changes progressively in time to generate various cell types. So now we can computationally infer differentiation trajectories from single cell data to generate a branch tree. So here you can see that the tree that we have generated reflects the differentiation status and also age of the cell types that are generating over time. And nicely, they can show also the markers, known markers, for instance, here for the apical progenitors and glia, and also intermediates and later excitatory neurons and other types of neurons that we have here. One interesting observation here is that, there has been debated in the field whether the progenitors has been fated. And in our tree, we can see that, for instance, neural population, all of them are coming from a shared molecular trajectory, means that they have a common progenitor. This is an important finding, shows that actually the projection neurons diverse post-mitotically, and the progenitors are not actually pre-fated. So you can also see nicely that from our tree, you can see the expression of known markers, for instance, like in terms of different markers for corticofugal and also callosal neurons, and also you can see it for other cell types like astrocytes and other cell types in the brain. Another important findings that we had here was that when we reconstructed the tree, we saw that, for instance, there is a gene called PCP4, which is a marker for corticofugal cells, also appears here in the intermediate cells. And actually, when we mapped it back into the topographic structure and see whether this is true in a spatial data using Slide-seq, we see that indeed actually PCP4 is expressed also in intermediate neurons, showing that these markers haven't actually been assigned to these migrating neurons in the past. Then another important aspect from this branching tree is that now you can go from the early time points to each trajectory, and then define the cascade of the genes that are getting an on and off during this process to see when these genes are expressed over time. And now you can do a lots of perturbations about it to see when the genes are important, when they get on and off during this process. Additionally, when you have such a trajectory, you can go now to any given branching point and find molecular codes of branching point transition. So here what I have done is that to find the top distinguishing features or genes or transcription factors between cells in, for instance, here in the siblings versus the parent branching point. I used and trained a classifier to distinguish a class, for instance, the parent versus the rest, and ask the classifier which genes or features are more important or more informative to the classifier for distinguishing each of these compared to the alternate branching. So this actually allowed us to identify candidates, like, each branching points, and come up with a set of genes that either we know it in literature, and also come up with novel genes that at each branching point perhaps play an important role in decision of that process. So this is perfect because, what we have done is based on single gene, but we know that usually in a complex biological process like corticogenesis, we can more robustly describe this phenomena by a set of genes that either co-expressed or, expressing at the same time. So we used approach called non-negative matrix factorization. It's a multivariate analysis in linear algebra. When you have your gene by cell matrix, you can decompose it into two matrices. One would be the genes by effectors, or the number of programs that you choose, and also you scored all those programs that you define them here, number of metagenes in each cell of interest. Then actually I defined all these modules individually in each time point, and identified gene modules across each of them, annotated them using the top 15 genes, and then chained the modules from consecutive time points to define genetic programs that represent different aspects of corticogenesis. For instance, you can see here we have broad terms like neurogenesis, but also we do see specific programs that coming up later on, for instance, around 30.5, again saying that they have a lineage, like a common lineage in terms of progenitor. And also you can see we have different types of neurons are coming up with the specific programs. Also, we can map now these programs that we defined based on this program scores, which is not one gene, but a set of co-expressed genes, back them into a Slide-seq data. And we can see nicely this is aligned with what we expect. Each cell type is expressing those type of programs in the spatial data. This is nice. This is transcriptional data, but when you're talking about transcriptional data, you always expect that there is a regulatory process underlying that one. So to gain more information about this regulatory process, deriving all these transcriptional changes associated with this branching tree or lineage segregation, we used single cell attack-seq at different time points. This is 3 time points, E13, E15, and E18.5. For each individual time points, we inferred the gene activity scores, which is just simply calculating the gene body and counts over gene body and promoter accessibility, and we used that one to combine it and integrate it with single cell RNA-seq to transfer all the labels that we define and identities that we define using single cell RNA-seq, and then by that we defined all the cell types that we defined in RNA-seq in the attack-seq data. So also to further check the consistency between this transcriptome and attack-seq data, we co-embedded single cell and RNA-seq using a shared UMAP. As you can see in the left panel of each of these individual time points, you can see that the data was very well merged, indicating that chromatin accessibility also captured the full cell type spectra identified by single cell gene expression. Then now we ask, okay, now if we go and reconstruct the same tree that we have established for RNA-seq data, do we see also the same structure and branching tree, or we do see, for instance, the prefated progenitors? Because we show that actually from the RNA-seq, you don't see this free prefated progenitors, but the question was whether that could happen from the chromatin or from the epigenetic landscape. So we use the ATAC-seq data gene activity to build up a developmental trajectory of cortical cells, and we found that actually a similar structure to the single-cell RNA-seq data. And also we do see that there is a unified actually progenitor deriving post-mitotically, and these are not prefated actually. And when we look at the markers, we can see that they are segregated as expected, but more broadly actually, which potentially means that they might be more priming and more plastic. And one only interesting thing that we captured was that these putative near projecting neurons were only actually the only population that was assigned in different branches in RNA-seq versus the ATAC-seq data, which means that these neurons may be more actually related to corticofugal and deep layer CPNs. Another aspect that we can see now from this is that whether usually this is a general idea in the field that chromatin accessibility preceded gene expression. So we check it for several genes, and we do see that there is chromatin accessibility is preceding the gene expression. This is the pseudo time, and also this is the gene expression level, and we do see that, and then actually suggesting that epigenetic lineage priming is happening here during this process. Another interesting aspect would be that now you can uncover the dynamic changes of regulatory interactions during the course of corticogenesis. Therefore, we generated a map of all predicted cell type state specific cis-regulatory interactions by calculating co-accessible sites with Cicero algorithm developed by Colette Ropnell lab. And we use this map to link differential accessible sites or enhancers or distal elements to a specific target genes, either over the whole process of differentiation and trajectory, or during for the later time points or the terminal time points that you can see here. Interesting thing here is that now you can go, and because this is a cascade of motif or accessibility over time, now you can go and do the motif enrichment to see over all these differential accessible regions, what are the transcription factors that are playing a role. Here I am showing across the tree, and also here for the terminal cell types. And also here we only look at those genes that has expressed in that targeted cell type. And for each of these genes, I didn't bring it here for the sake of time, but for all these transcription factors, we validated with Slide-seq data, means that each of these motifs that are enriched at a specific time point during the either migration or during the generation at a specific layer, we also confirmed all of them that are expressed at the specific place using a Slide-seq data. We defined, for instance, here, some of this has been reported in the literature previously, but also we found a couple of new transcription factors that sounds playing important role either during the migration and also for other cell types that has been generated here. And another interesting things about PCP4 is that there is an interesting example. There is a regulatory element that target PCP4, and these elements actually, there are different enhancers that we define that, and these enhancers actually occupied by different transcription factors, because this is different differential enhancers that are either expressing in E18 subcerebral projection neurons or in the migrating neurons. And we identified these distal elements in both like E13, E15, and E18, suggesting that cell type specific enhancers present during like this transition, and they are actually occupied by different transcription factors over time. For instance, in subcerebral projection neurons, we know that this is occupied by FESF2 and BCL11B, and in like earlier stages, we do see that this is, for instance, only by BCL11B. Now, this is interesting because now we have all these single genes, we have all these like programs, and the question is that what we can do it. Can we test these regulatory code or modules to see whether how functional and how important they are? So characterization, I think loss of function, a mouse model has been, we know that there were several genes in cortical development that has been, highlighted over the couple of decades. But the vast majority of these efforts had to actually was a kind of trade-off between like the throughput or cellular resolution. I think single cell technologies enable us to rapidly phenotype mutants in an unbiased manner and look at the specific identity and characterize that actually will be generated after a gene or a set of genes get mutated or knocked out. So to test this, we used FESF2 because FESF2 came up several times over during our analysis. We showed that during like this branching tree play an important role during this process at the branching of separating callosal and corticofugal, and also we showed that this is coming up early on one of these gene modules that we found as one of top ranking genes. So there is like a question, because we know that when FESF2 get knocked out over like by several labs during the past studies, it caused a complete loss of subcerebral projection neurons, but the mechanism behind it and the identity of the neurons that are produced in their press was poorly understood. So what we have done here, we have knocked out this gene and collected the data from E15 and E13 mice, and we look at the single cell data. Now we defined all the modules in the knockout and also the heterozygous, because we didn't have the wild type from the exactly genotype background, and we compare it with the whole modules that we define in the wild type data, the big atlas with 100K cells. And interestingly, we find all those modules, and we can see nicely that when you knock out FESF2, all the genes that we define on that modules get like knocked down and down regulated. And interestingly, we know that we find one module that wasn't actually present in the wild type data, and that module was enriched actually for axon development and gene guidance, consisting with the fact that there is an aberrant axonal projection in this mutated cells. And now, because we have this big single cell data set, we can train a classifier and ask, okay, what's the identity of these subcerebral projection neurons that, you mutated FESF into that, and we don't know what's their identity, what kind of identity now they acquire. So we look at that, and we see that based on the classifier, half of those population get clustered as subcerebral projection neurons and corticothalamic projection neurons, but their identity is different from the wild type corticothalamic projection neurons of subcerebral projection neurons. Means that actually, based on what we have analyzed with details, shows that actually you need FESF2 to suppress, for instance, the genes that coming up from the subcerebral projection neurons identities. And as a summary, we here, generated a comprehensive single cell transcriptional and epigenetic atlas of the developing mouse somatosensory cortex. We captured the development of all cell types throughout mouse corticogenesis and identified longitudinal molecular dynamics that accompany lineage specification of individual cell types. We identified candidates, molecular effectors and regulatory elements that underlie phase divergence. We actually validate them based on spatial data and also some fish data. And our molecular map now enables the mechanistic understanding of aberrant corticogenesis. By that, I would like to, thank my advisors, Aviv Regev and Feichen at the Broad Institute, and also Paula Lota and Daniela Di Bella at Harvard Stem Cell Department of Harvard Regenerative and Cell Biology, and also thanks to Evan McCusker, Robert Stickles, that was the one that developed Slide-seq, and other people in the lab that helped to, make this fantastic work possible. Thanks.
Thank you, Ehsan. That was a very nice talk. Just to remind everybody, if there are any questions that you have for us, just start typing them into the Q&A section below. There's already one question. But if I could start off, actually, Ehsan, one question for you. So, biology is very dynamic, obviously, as you demonstrated here and everybody knows, and you were able to elegantly capture the development and progression of these cell states over time. But, and you have validated the model with your knockout, but what are, in your view, the next steps for what this means for the biological community in general? Because people can always identify cell states, cell types, and map them, but like, what are the real world uses of these types of technologies? And
I think the most important part would be that, because we could go with high resolution with single cell data, but we lose some positional information, which is important, right? And we try to kind of complement that one always with Slide-seq or other types of fish data. So I think having an unbiased way to be able to capture both spatial with the same resolution at single cell RNA-seq that we have would be actually a plus in the future. And another thing is that now we generated all of these programs and elements that now I think provide a way to go and start to perturb that, to see really how much of those programs or elements that we have generated based on this single cell actually would matter during differentiation of these cell types, and the sequencing and timing generation of these cell types, how much they're dependent of all those transcription factors. Or so I think it generates a lot of opportunity to start doing perturbations. Either that would be a kind of in vivo perturbation, or actually other model systems do that.
I see. Okay, makes sense. Okay. Some questions. There's one for Ehsan. How did you identify the enhancers, or were they already known?
Yeah, so we actually used ATAC-seq. So we first come up with we combine them, make them pseudo bulk to be sure, because we know that the depth of ATAC-seq is not that high. And also then we used we defined each element, so we call the peaks, and in each of them separately in a pseudo bulk way. And then also we used a C0 connectivity map to connect different enhancers in cell type specific way. And then by that, actually we defined all the elements. And also in that way, we removed all the promoter ones, because we know that the enhancers, we just wanted to focus on distal elements. So we should remove the ATAC-seq peak or binding sites from the promoter to be sure that we are focusing on only only distal elements. And then now, like, you know, we have started to do other types of like differentially ataxic binding sites to define only those sets that are matters over the dynamic process, and like do the motif enrichment over those.
Okay, thank you. Okay, so a question for me is compatibility with fixed tissue. This is a common question that we get all the time. A lot of people ask us about FFPE or just fixed tissue in general. This is very high on our list, and you know, we are going to be working on this in the future. There isn't really any timeline of when this will be available, but you can imagine this is something that would be very high on our priority list. But you know, in the meantime, you know, with a very small team, we're trying to focus on the foundational technology, making sure that the beads are very robust, the assay is very robust, before we really tackle those omics, other omics, I guess. Well, it's not really omics, but you know, different formats of tissue and so on and so forth. As for BCR TCR, as some of you may have already seen, there is a slide TCR-seq paper already in the literature from, I think, last year or the year before. You're welcome to try it. Theoretically, it should work on our current platform, but we haven't developed any off-the-shelf kit for that yet. But again, that's something on our list to do. Okay, so that is done. Price of the 10x10 versus a 3x3. Just for the kit itself, the 10x10 will be more expensive, but if you calculate it per unit area, it's actually less expensive. So if you're interested in pricing, again, just contact us at sales@curiobioscience.com, and then we can give you details on that. Which sequencing platform can be applied to the libraries? So what we outline in the user guide right now is for Illumina sequencing. We do specify using the Nextera XT kit for the fragmentation library prep at the end. We've had several customers that are interested in using other sequencing platforms, but that's not something that we can tackle right now, but they're trying to get it to work on their own. We do have some documentation that will show you the actual sequences of various steps within the protocol, so you can figure out how to get that to work if that is something of interest. I know some of these newer sequencing platforms do have conversion kits, so that's always an option if you generate an Illumina library first, but you'll have to go to them for guidance. Next question, I believe, is for you, Ehsan. So to run your analysis, did you include all RNA species, including non-coding RNA and microRNA, or did you work on mRNA transcripts only? You're muted, by the way.
It was mRNA transcripts only.
Okay.
But I think also this could be relevant also to, well, that's poly-DT. So you only capture in the Slide-seq, you only capture mRNA, not other types of, but in our case, for single cell data, we just use the mRNA transcripts.
Okay. You envision non-coding and microRNA being useful in the future if this were enabled? Yeah. Okay. Cool. There aren't any other questions. One other question I had actually was, you guys use Tangram. Obviously, there are other integration pipelines out there like cell-to-location, RCTD. So in your view, like, do you have one preferred method, or is it just really dependent on what you're doing? Are they some better than others in certain circumstances? I'm sorry, you're muted again.
Sorry for that. We used Tangram at that time, but I think RCTD works very well because, like, also, specifically also because I know very well in the lab because it has been developed in Phase Lab. So I think different versions of RCTD now, they're does a great job actually, and also there has been a benchmarking paper out that compare all of those, and RCTD was one of the top ones in terms of like do the transfer mapping and like decompose the data.
Okay, great, thank you. Yeah, so last call for questions. Okay, so could you introduce a bit about the bioinformatics analysis pipeline, any knowledge and program required? Okay, so I guess this is for me. So there is, so like I mentioned, there is an online version right now, or online downloadable version that you can install locally, and we can help you get that installed. And this is just to convert the FASTQ files you get off the sequencer into matrix files, right? So there is, one of the outputs is a Seurat file. So a lot of people use Seurat for single cell and also spatial. So if you want to use that, you can use that. But there are also raw matrix files that are output that you can use in any secondary analysis tool that you want. So at least for the secondary analysis, you will need some programming knowledge or be in touch or working with other bioinformaticians who can do this very easily. So, nowadays there's a lot of bioinformaticians who know how to do this, and so that should be pretty easy. So, that's what guidance that we have. We can, if you have any more questions on this, we can always provide guidance internally. So we can help you with at least pointing you in the right direction for secondary analysis pipelines that you can use.
And also just add on top of that, like for instance, RCTD that has been developed in Phase Lab also work very well. It's an R package that it's very user-friendly, and you can use it for a lot of post-processing analysis, like, differential gene expression and so on and so forth. So that's another one that you can use.
Mm-hmm. Yeah, okay. Well, if there are no more questions, thank you, Ehsan, for joining, and thank you everyone for attending, and we'll see you all next time.
Thank you very much. Thanks.
Thank you, guys.
Bye.